A lot has to go right for animal life to come into being, let alone live for any extended period of time. And even if things do go right, a lot can go wrong. Today we'll be looking at type 1 diabetes, specifically how things can go wrong on the cellular level that lead to the development of this condition. Remember, we're all students here, and we can all learn from one another. Enjoy. Generally speaking, type 1 diabetes is a disease of the pancreas, specifically pancreatic beta cells. Beta cells are necessary to produce insulin, and insulin is necessary to transport glucose into the cell from the blood. If beta cells are dysfunctional or destroyed, the glucose cannot get into the cells. A condition that results from this is known as ketoacidosis, which is when the blood becomes dangerously acidic due to ketone body presence in the blood because that glucose cannot get into the cells. While this condition can be treated with insulin injections, for instance, it can be fatal if untreated. It's a very dangerous condition. Now, I want to discuss some of the mechanisms for which beta cells can be affected and insulin can be affected. As depicted on the diagram on screen, one mechanism by which insulin production is affected is due to the production of autoantibodies against insulin, glutamic acid, decarboxylase, and even beta cells themselves. However, I wanted to go into much more detail about some of the lesser known mechanisms in the development of type 1 diabetes in the upcoming slides. A few mechanisms outside of autoantibodies that affect beta cells in type 1 diabetes are seen here. We have endoplasmic reticulum stress, we have oxidative stress, and we have high vascularity. First, I want to briefly touch upon high vascularity, and that is the most intuitive. High vascularity increases the access of autoimmune T cells to beta cells in the pancreas, destroying insulin and destroying beta cells. These next two sections I want to go into much more detail. And the first one of that is endoplasmic reticulum stress, what goes wrong and how it contributes to the disease. Now, normally the endoplasmic reticulum is where folding takes place of proteins. If proteins are backed up and not folded properly in the ER, the unfolded protein response, or UPR, is initiated under normal conditions. The UPR is made up of three proteins: IRE1, PERK, and ATF6. These proteins either ensure the unfolded proteins overwhelming the ER are degraded or that they are folded properly. However, it is possible for the ER to become damaged or dysfunctional. This could be due to, for instance, inflammation through the release of toxic cytokines or oxidative stress, which I will go into later. In type 1 diabetes, this kind of damage is very common. It contributes to the inability of beta cells to either properly fold or produce insulin. Endoplasmic reticulum stress due to a dysfunctional UPR is so disruptive to normal insulin production that the loss of function mutations of the protein PERK in the UPR alone is enough to develop permanent insulin-dependent diabetes. Next, let's take a look at the effect of oxidative stress on the destruction and dysfunction of beta cells. When beta cells are exposed to oxidative species, whether it be organic, radiation, or a molecule like hydrogen peroxide, it can be damaged or even destroyed, leading to decreased insulin production. This occurs because oxidative species can damage DNA through the oxidation of thymine. If a cell cannot handle the oxidation, it may signal cell apoptosis, or cell death. This is especially a problem for beta cells, as they are particularly vulnerable to oxidation due to low levels of antioxidant enzymes. These oxidative species can even cause endoplasmic reticulum stress, an inflammation which leads to cytokine production. This is evident by different UPR genes being upregulated when exposed to oxidative species. Which brings me to the last thing I want to bring attention to. There's a lot of interplay between the various mechanisms of beta cell destruction. Endoplasmic reticulum stress itself causes beta cell destruction, dysfunction, but also is pro-inflammatory. Oxidative stress is pro-apoptotic, but it can also cause stress in the endoplasmic reticulum, as well as inflammation and cytokine release, which then in turn is pro-apoptotic. Inflammation promotes endoplasmic reticulum stress, and increased inflammation promotes increased vascularization, which itself promotes increased inflammation. So these different cellular bases of diabetes don't act in isolation. When something goes wrong in one area, it can even cause a cascade of beta cell failure by other mechanisms influencing each other. So putting it all together, type 1 diabetes is caused on the cellular level by the destruction and damage to beta cells in the pancreas. These beta cells are necessary for proper insulin production, which is necessary to get glucose into the cells. Beta cells damage or destruction can be caused by a variety of mechanisms, including autoimmune T cells, increased vascularization, increased inflammation, endoplasmic reticulum stress, or oxidative damage. And these different pathways by which beta cells can be destroyed all amplify the negative effects of each other. With that, I hope you learned something today, and remember, keep loving and keep learning. Thank you.